Hello, everyone. I'm Harshit Sahay, a PhD student in the Gordon Lab at Duke University. And today I'll be talking about assessing the enrichment of somatic mutations in transcription factor binding sites. Most somatic mutations in cancer are found in non-coding regions of the DNA. However, studies with large sample sizes have failed to find more than a handful of non-coding driver mutations. Part of this is because generating background distributions for mutation rates in these regions is much harder than in coding regions. Several processes can contribute to mutation rate variation in regulatory DNA. So here are some of these processes that can vary on scale, shown on the x-axis, and also in their strength, shown here on the y-axis. Recently, a lot of work has pointed to transcription factor bindings such as CTCF and ETS and other factors at promoters as contributory mechanisms to mutation rate variation at the local level. Typically, such work points to the depletion of repair at these binding sites that causes an increased mutation rate in the binding sites compared to their flanks. Expectedly, this mutagenesis can be tumor-specific, such as AR here, which shows an enrichment in prostate tumors, but not in other cancer samples. And several studies have now seen this effect for individual TFs in specific cancer types, but a lot remains unknown. These studies propose competition with diverse repair pathways, but little is known experimentally about this. Also, this effect is tumor-specific, like we discussed before. As a result, a comprehensive evaluation of such enrichment is needed for many TFs at scale. Specifically, we are interested in analyzing mutation patterns in TF binding sites across different cancer types. Also, because of the lack of experimental knowledge currently, we're interested in focusing on testable hypotheses that provide a pathway to future experimental validation. In order to do this, we first need principal ways of identifying active binding regions that can be scaled to different TFs in different tumor contexts. With that in mind, we observe that direct binding data, which is ChIP-seq, is usually not available for most tumors and is only available for some TFs in some specific cell lines and tissues. In such cases, whenever this data is not available, we turn to indirect methods, where we can consider cases where we have some chip for that transcription factor, but not in our case of interest. In such a case, we combine all other ChIP-seq data across all other biological contexts and intersect it with chromatin accessibility in that tumor and tissue. The reason this is scalable across different tumors is because accessibility data is usually available for most cell lines and tissues, and also for several tumors, unlike ChIP-seq for most factors. In cases where there's no chip available at all, we use motif calling tools to directly identify sites in accessible DNA. This shifts the problem to deciding what the right accessibility data for a given case is. Several factors can contribute to this choice. Transcription factor binding evolves as the cancer evolves, shown here in this study. This means that accessibility in normal samples, which are tissues, and cancer samples is just a snapshot at either end of this process. Additionally, the data that usually exists is in the form of DNA-seq for tissues and ATAC-seq for primary tumors, meaning that these datasets can vary both in the biological sense, where the tissue and the cancer are different, but also in the experimental sense, then these are profiled using different techniques. We first want to better understand this variability. To explore this, we check accessibility datasets in the same cell line that we matched for coverage. Despite being measurements of the same context, these datasets differ significantly. Shown here is an example of this for the LNCAP cell line, where we took ATAC and DNA-seq samples matched for quality and coverage, but found that only a third of the peaks intersected between the two datasets. In such cases, we need to figure out which group is the right group to use, or which dataset is the correct one to use. For our case, since we're interested in identifying active binding regions, we check for how these overlap with TF binding data. Performing this analysis of CTCF, we see that the intersection region, that is the peaks common to both these methods, shows the highest overlap and is thus the most enriched for transcription factor binding. This is also true for other cell lines as shown here, and also for other factors as shown here for MYC. This suggests that the intersection region is more likely to be enriched for active binding regions. Moreover, this also suggests that measurements in the same biological contexts can vary significantly, underlining the importance of making the right choice. Now, we want to look at biological variability, i.e., variability between attack-sequence DNAs now coming from tumors versus the matched tissues. Performing a similar analysis, we looked at overlap for each of these sets with CTCF binding data. In each case, we took an attack-seq set for a primary tumor of interest, shown here on the x-axis, and matched it with the relevant tissue type or cell line which had DNase data available. As we can see in this case, our comparisons are affected by depth. So between attack and DNase, one of the two can outperform each other based on which tissue type and which cancer we look at. But across these cancers, the intersection still has the highest enrichment for transcription factor binding. With this in mind, our general strategy, whenever attack-seq from primary tumors is available, is to use only the intersection with tissue-specific DNase to identify transcription factor binding regions. Whenever this data is not available, we default to the use of DNase from the tissues. As a case study, we turn to CTCF, a transcription factor that is highly expressed and likely to be present in several tumors. It is also shown to be enriched in several tumor types. More importantly, CTCF has CHIP data in almost every tissue, which means that we can compare and contrast our direct and indirect strategies, and also responds best to PWMs for accurate site calling. We first look at this direct strategy shown here for esophageal tumors. To recall, the direct strategy means we directly take ChIP-seq data from all esophageal tissues and cell lines and call binding sites within these regions. Once we profile these mutations, we see an increased mutation rate in the regions bound by CTCF, shown here as in red compared to the flanks. CTCF often binds with the cohesin subunit as seen here, and zooming in, we can see that this enrichment typically comes from mutations both within the binding site, shown in dark red here and underlined in black in this motif, as well as mutations at the flanks of this subunit, shown in lighter red here and in the extended motif here. Together, these mutations contribute to an increased local mutation rate. We next asked if our indirect strategy could also recover this pattern. As a reminder, here we take the union of all ChIP-seq data sets apart from those that are in esophageal tissues and perform the union that gives us all ChIP-seq data observed for CTCF. Next, we intersect this with accessibility specific to esophageal tumors or esophageal cell lines, giving us an active binding region shown here in orange. Profiling these, we again see a similar pattern, suggesting that our two strategies are consistent and that our indirect strategy can be used whenever ChIP-seq is not available directly. Looking at other tumors, we find that this enrichment is specific to tumor types. So, for example, gastric cancers here show the same pattern of enrichment as found in esophageal tumors, whereas brain tumors show a slightly different pattern. On the other hand, kidney tumors show no enrichment at all. This suggests that the mechanisms behind these mutations are likely specific to tumors and need to be analyzed separately. To move from an enrichment to a mechanism, we use mutational signatures, each of which represents a different mutational process. We take all the mutations observed within binding sites and flanks and attribute each to a signature, finding that signature 17 contributes to the largest amount of mutations in these regions. Indeed, profiling signature 17 mutations in specific, we recover the same pattern within the binding sites as shown in the larger data set, suggesting that most of these mutations in binding sites come from signature 17. This is consistent with what has been implicated in other studies in colorectal, gastric, and esophageal tumors. However, the etiology for the signature remains unknown. Looking at other signatures, we see some mutations from signature 1. While these don't drive the enrichment patterns we see, they do have a defined mechanism. This moves us to a testable hypothesis where these mutations in signature 1 and in binding sites can come from a TG mismatch that is created upon deamination. While typically this is repaired, a transcription factor binding could impede this and compete with the repair enzymes, leading to enriched mutations. Additionally, most cancer somatic mutations are actually attributable to DNA replication errors, which are also repaired by different repair enzymes. However, to test mutagenesis here, we first need to know whether these transcription factors bind the mismatches in the first place. Previous work sheds very little light on this topic. To address this, we've recently developed a high-throughput chip-based assay to test binding of transcription factors to mismatched DNA. We found that this binding was very different from that predicted by Watson-Crick DNA, suggesting that existing data is insufficient to explain this binding. We published this data set for 22 transcription factors and explored the structural aspects of this binding in our work. This work gives us a map of recognized mismatches for 22 transcription factors and allows us to test whether mutations coming from those specific mismatches can indeed lead to mutations after competition due to competition with repair enzymes. And to summarize, the goal of our work is to analyze mutation patterns in binding sites with a focus on testable hypotheses that can be validated experimentally. Specifically, we discuss how to build robust mutational profiles by using intersections of data sets and how to tie these to specific signatures and specific mechanisms. By focusing on highly expressed transcription factors to tumor type identified from RNA-seq here, we can build these profiles for both tumor types and TFs, and whenever possible, compare and contrast our high throughput data that gives us testable hypotheses for competition with mismatch repair enzymes. And with that, I'd like to thank the committee for giving me this opportunity to present, and also my advisor, Dr. Aluka Gordon, as well as members from my lab, both for inputs on this project and in collaboration in general. I'd also like to thank members of the Al-Hashimi lab who are collaborators with us at Duke, and members of my committee, as well as my department at Duke. Thank you.
Well, thank you again for such an interesting talk. I'm going to wait for questions, and I'm going to start with the question. So why do you think there's such big differences between different cancers? Do you think it has to do with the DNA repair that's happening and that's specific for the cancer, or with the treatment, or are there any other reasons?
Yeah, so I mean, part of it is driven just by the different mechanisms that are active. So in some of the cancers that we discussed, the prominent signatures that we see are just signatures that aren't active in other cancer types. So it's probably coming from completely different mechanisms driving most of the mutations. The other part of this would be basically the differences for that transcription factor binding itself. So it may or may not be expressed in some other tumors to the same extent, and hence not be available for mutagenesis.
Oh, so you haven't actually looked at what could have been mutated. Sort of, it depends what you're looking for.
Right. So we first identify binding sites and then look at mutation patterns. So if that specific transcription factor is something that's lowly expressed, we sort of do this only for highly expressed TFs. But if theoretically you were to look at a condition where it's lowly expressed, you might find lack of that particular enrichment in a specific cancer.
Yeah, that makes sense. But the CTCF, you would assume, is in all tumors, right?
Right, yeah. So for CTCF, I expect that most of the differences are coming from the signatures. So every tumor that has signature 17 shows that specific pattern, and sort of the other tumors that don't. So it's while we don't know what that signature does, it's probably the one that actually interferes with CPCF binding somehow. So.
Mm-hmm. Okay, so I don't see any questions, but I thought it was a really interesting session, all the way from designing mutations to looking at natural mutations, or mutations in cancer, and you're looking across transcription factor binding and RNA structure. So I would like to thank all the speakers for a wonderful session, and I'm going to turn it over to Jian Ma, who's going to introduce the next keynote speaker.